Good evening, everybody. So I hope everybody's have already run the course which was given yesterday. So I will just continue from the next course. Okay. So till this code we had run yesterday, and we had seen the length and the contents of this particular code, that is particular data. Is everybody with me in the same page in with the same codes?
Yes, sir.
Okay, Pratima,
Yes, sir.
Varshini,
Yes, sir.
okay, Osuri,
Yes, sir.
okay,
Yes, sir.
okay. Now we will go ahead and first we should load the library of chipsec. That will also take some time. So as I had told in the last class, that already this BIOS manager is already installed for you. So directly you can start writing with the BIOS mini manager install chipsec. The library which we will be using now is the chipsec. Okay, as I told, this is again a bioconductor package. So you need to install with the BIOS manager and library chipsec. Okay, please do it. It also takes some time. So we will go ahead with the preparation of the chipset using with the control samples. And we need to estimate the mean length using the estimate mean I will be giving in this particular in the succeeding codes. Okay. For anybody, this chipsec library is installed. Please let me know. Varshini, Raghavendra, Pratima.
Not yet, sir.
Okay. Osuri, Hamsini. Okay, please do it. Because the NGS experiments, it takes lot of time, and the main time goes with the installation of these bioconductor packages. If usually with the CRAN packages, it usually do not take much time, but the bioconductors, it takes some time. Anybody for whose it is executed, please you can unmute and let me know. For me, it will take time because my internet connection was not there for some time, so I could not load all the codes. But all the codes are already executed for me. I kept ready, so no need to worry on that. Please let me know if anybody's execution is completed. Completed for anybody? For anybody, it is completed. Pratima, Raghavendra, Pratima, Raghavendra, Varshini, Osuri, Hamsini. Okay. So for me, all the previous codes are completed now. The codes of yesterday, now it has started this chipsec. So why I did not tell, you can see this all packages are coming with the chipsec. Okay. So from now onwards, you are actually taking this particular chipsec library and executing the data as per the chipsec analysis. So anyway, in the meantime, I will also give you this particular codes and I will explain to you. So you can see now we are preparing the chipset. Already you have given the median. Median, everybody knows it is the mid value for the estimated mean of fragment, that is the reads. For all the reads, you are taking the mean and median for each. Then your cat means, I think everybody knows cat is the joining operator. So you are fixing, and along with the cat, the paste function is needed, because paste means if there are two strings, so it will directly paste it. So you will get the output like median fragment size for this library. Along with this, you will get the fragment length, and then you will, and along with that, it will come resize with all the reads, and it will return the trim for the reads. Okay, this is what is the particular function it will do. So you can execute anyway. It will take this will not take much time, but without this execution, it will not happen. This will hardly take a minute if I run, but only after execution of this one. So the output you will get like this. Anyway, it is executed for me. You can see here, you will be getting like this. So I have removed the output. It will come here. I will show you. Is it executed for anybody? Hamsini, Osuri, Pratima.
It's taking a while, sir.
Okay. Raghavendra?
It is still running.
Yeah, for me, even this step is executed and this is also executed. So I hope it should be executed for everybody because for me it was running. I think you all did not execute the steps initially. For me, this is also completed. Pratima, Raghavendra, Varshini,
Yes, sir.
done?
Yes, sir.
Okay, great. Others? Saida, Hamsini? Okay, let us go further. So now we will apply with our chipset sample already we prepared initially. That is in the yesterday's class, sorry, day before yesterday's class. So you can see that the input which was already taken, that is prepared our chipset. Or the we are making the function and we are preparing our input as per that. Then for the rep one data which was prepared last day, for that we are implementing this along with the rep two, so that you can see the rep one. For also you can also see, if you want, you can see the other value, that is input and rep two. So it will hardly take a minute. From now onwards, it will not take much time. Only the installation takes time. So you can see here directly all you are getting directly the samples and the I ranges already I told, and you are getting directly the scores for each one, which is for the rep one particularly. Okay. So let us move forward. So now we'll go ahead with the binning. So what is the binning? It is given here. That is the established genomic length. Okay, then we will be generating the bins. Okay, we will tie the genome into the non-overlapping bins of size 200 BP. To this, we need the information about the chromosome size. Already, you know the chromosome size is the chromosome is 6. Everybody knows it, and it is the mouse genome. Already in the last class I had shown you. Okay, so let us continue with the code now. So this is the data. So what you are getting here, so you are getting the information of this particular SI. SI is the data we are using here as the reference, and you can check it out. The particular, this is also of the MM9. Okay. So let us go further. So next we use the title genome function from the genomic ranges package to generate the G ranges for the intervals. G ranges, I think everybody knows, it's the genomic ranges, okay, of the 200 BP. So we can see the bin size I have given here as 200, and I have selected the chromosome 6. Okay. So what do you see here? Mainly only the genome ranges are given. The chromosome 6, because your chromosome for the particular input which is given is chromosome 6, I ranges are given, and the strand information is no more needed in the further experiments. Okay. So next we apply to the objects of the input, that is our data which was initially taken, input REP1 and REP2. Okay, we obtain inputs of 200 bins for the input, for rep1 again 200 bins, and rep2 200 bins, which are the G ranges of objects. So we'll see it now. You can see here each and every codes are interlinked. So here you have given, see, input of the bins. Bins is defined here initially. Bins, you can check it out. So for again the bin chipset function, it is generated here. So using this, you are getting the input for the 200, which is already defined initially here. Okay, bin size is 200. So that is for the, we are taking the output of only this REP1. Okay. So we will now generate a plot. Okay, so you can see here in the plot it is given for the X lab it is chromosome 6 and for Y lab it is counts per bins. So for each and every bin you can see how many how many reads are there. It is given here. Okay. Now if you tell sir in the R this picture was, we can take it even here also it is possible. You can see the option, right click and you can see you can save the image. Okay, here PDF option is not there as per R. So R is little better, but this particular platform is needed. I told you yesterday was the Linux platform is needed for executing this particular codes. Okay. So next we need to export the binned data. So at this particular step, the data is ready for the visualization. One of the most common means of the sharing chip data is to generate the week, bin, and bed graph file. So we will do it now. So this we need to install this package. It will not take much time. This will be useful for the visualization. Okay, now we will use go ahead with the visualization. So the principle of working with this GVs relies on the generation of the tracks. You will be seeing several tracks on the once you are seeing the visualization for the chipset, the chipset, the data which we will be getting it. So sorry, not the data, but the output. Okay, so you will be seeing the chipset peaks. That is what I had told in the lectures. So peaks are important. You will be seeing the peaks in this particular codes, the gene models or any kind of other data as annotation for the CPG islands. I think CPG island you might have studied in the class. Okay, we start with the loading of the gene models for the chromosome 6 because already you know is that your the data is taken from the chromosome 6 position, and the position was displayed initially the same one which is given here. Okay, we focus this region as it harbors the nanolog gene which is strongly expressed in the ES cells. Okay, so coming back, let me see if it is done for me. It is still executing. So we will take the BM data, that is which contains your reference genome. I hope everybody is in line with me. I did not check anybody's. Pratima,
Yes, sir.
at which step are you in? You are getting the results?
GWIS installing it. It's executing still.
Sorry?
I'm installing GWIS. It's not run
G
yet.
is okay. Osuri? Sanjana? Why all are silent from I think from last 1 week? Apart from Pratima, I am not able to get response from anybody. Varshini?
Sir, I'm installing that GWIS step, sir.
Okay.
Like that.
Osuri, are you present there?
Sir, I'm installing Sir GB.
Okay.
I'm trying.
Okay. Osuri? Saida?
Yes, sir, I'm also installing.
Okay. So for me, this is done. So now we'll import the data. You can see it is the, I think I shared this code, yeah. So I think you can check it out. It is the gene region is RefSec, it is the reference sequence. Genome is MM9, active chromosome is 6, and the annotated features is 102. So we'll see this now. So we included the genome axis track object, which is the coordinate axis showing the genomic span of the analyzed region. And now we will be plotting the result with the help of plot track function. So we choose the region into the region to zoom into which to and from the arguments. And this plot you can check it out. One moment, I will give you the code, so you will check it out. So first thing you can see here, this is the plot of the reference sequence. Okay, do not get confused. You can check it out here. This is your reference sequence, and the nucleotide you can see here 222.7. So finally, we will plot these tracks along with the genomic features, and we will observe the uniform coverage in the case of input tracks and the pronounced peaks in the enrichment of the H3K227AC. So we will check it out with the codes now here. So this will be your peaks. So you will be getting the peaks for your data. You can see here, this is your input, this is your rep1, this is your rep2. Okay. So now further analysis we will be doing in the succeeding classes, and we will go ahead further with this particular work. So already you have got the chipset peaks. Now we will go ahead with further with the analysis of this one. Okay. I hope till this part everybody has done. Pratima, Varshini,
Yes, sir.
Varshini done.
Yes,
Did you
sir.
get the peaks?
Yeah, sir.
Sayeda, Sayeda, Osuri.
Yes, sir.
Okay. Can anybody share the screen? Varshini, can you share? Before concluding, I just want to see everybody's work. I think can.
Sir, you have to stop sharing your screen only, then I can share.
I think I have already permitted. I think. Just try to do it. I already, one moment. It has become different. Yeah.
Can you see my screen, sir?
Yes. Yes. Very good. You have already got the peaks. Okay. Pratima. Pratima. Okay. Fine. You got the peaks. As per if the type you can see it is histogram. Okay. So histogram, you can see now everything is given there. Type is histogram, sys track is 0.7, font size is 10. So as per that, your plot is represented. Saida, Saida, can you share the screen?
Sir, I do class on my phone and do it on my laptop, sir.
Okay. Okay. So I will see you tomorrow at 6 o'clock. Is it fine with everybody? Tomorrow evening, 6 o'clock. And as I told today also, everybody please run all before joining the class. Okay, so is in the sense, so it will not hamper your works, and you can be along with me during the class. Okay, so please do this, everybody, and be present by 6 o'clock tomorrow. Okay, good night, everybody. See you.